Hey class, today we're going to get started with some DNA microarrays. So DNA microarrays are kind of older experiments or platforms that would measure the expression of a gene by measuring the amount of mRNA that has been transcribed by that gene. It's kind of moved aside a little bit towards Rna-Seq, with Rna-Seq becoming cheaper and cheaper. There are a lot of advantages to Rna-Seq over microarrays. So you may ask, why are we doing this? Well, the reason is there's a ton, just a tremendous amount of data available, public data of microarray experiments. And so there's still utility, especially in bioinformatics, where we do these meta-analyses where we look at publicly available data and take large amounts of data and process it and see the commonalities between experiments and things like that. There's a lot of use still for that microarray data. Just because there's a new technology out there that has kind of supplanted it doesn't mean that we should cast it aside and not use it anymore. Going forward, if you were to design a new experiment, I would definitely use Rna-Seq, because the benefits of novel transcripts and unknown genes and things like that. But there's still a lot of utility in microarray and knowing how to process this older data. So today we're going to get into how to do microarray experiments and the kind of utility that they have. But first I want to show a way of accessing some of this public data. So I work with NASA GeneLab. So NASA GeneLab is a data repository for spaceflight or NASA related data, or data of interest to NASA. So I know the people that run GeneLab. They're awesome people, and they've designed a really good repository. I'd argue the best data repository for next generation sequencing data, because it's well curated, it's specific, and it provides some other benefits like intermediate files, which we'll use in the Rna-Seq stuff. These are data sets that are publicly available. We have GLDS, which is the GeneLab data set, and then the number. So you can see that currently there's about 467 data sets on the GeneLab repository, all dealing with something to do with spaceflight. So whether that's experiments that I've conducted on the space station, so how plants grow in microgravity. Here we see a Rna-Seq experiment on bone isolated from hind limb unloaded samples. So what they do is they put these mice in for 11 days in a position like a mounting where they don't use their hind limbs for 11 days, and they look at the expression changes. And the reason they do that is because in space, since there's no gravity, there's no real weight being put on the muscle, so we see a lot of muscle deterioration in space. This data set looks at that. Here's another one. The effects of spaceflight on eyes, specifically the arteries, veins, and lymphatic vessels. You can see this is the organism is a mouse. And so there's a lot of really good data here. We're going to look for BRIC16. This was a study done by my previous postdoc advisor. So there was 3 BRIC16 experiments. But when I type BRIC16, his comes up. Dr. John Kiss, who is the Dean of Liberal Arts at UNC Greensboro, great researcher. So I'm going to pull up this data. And as you can see, there's a lot of good information here. So the context, so if you have questions about the data set that's in this repository, you can see that Dr. Kiss is at UNC Greensboro. They give his email address. They tell you what the samples were exposed to the factors, so space flight, so microgravity, but it also includes anything that is encountered in space flight, and cosmic radiation, which is kind of part of the space flight. It's a transcription profiling experiment, so you're looking at the profile of transcripts, so the amount of mRNA and how it is different between ground and space. It's a DNA microarray. The device platform, there's different microarray companies. Affymetrix was probably the biggest or most used platform. In this, depending on what platform you're using, you would run the analyses differently. It gives you the title of the experiment, the type, the flight program. So this was done on the shuttles. This was done years back. So STS-131. We know that STS was a shuttle mission. It was performed in a brick canister, which is just like a big metal tube. It was done through NASA and Kennedy Space Center, and where it was funded by. They give you acknowledgments for who helped out in the experiment. And then all the protocol too, which is great. So the growth protocol for how they grew the seeds, how the samples were collected, how the RNA was extracted, how the probes were labeled for the experiment, how data was collected, everything. So really well curated, like I said, compared to some of the other databases. Then they give you the samples. So as you can see here, in this experiment, there were 6 samples. We have 1, 2, 3 flight samples. So these 3 samples went up on the space shuttle. And then we have 1, 2, 3 ground samples. So if we're comparing spaceflight to not spaceflight, then you have to have a control group, right? And that would be the ground samples. And so they were grown in brick tubes just on the ground. We got our species, Arabidopsis, which is just a little model plant that we use for this type of stuff. All the data, how they were frozen afterwards to kind of take a snapshot of the expression, because if you don't freeze them while they're in space, then they come back down to the ground and get exposed to microgravity or to regular gravity, and then therefore you probably lose whatever gene expression effects that you would have. So yeah, a lot of great data there. These are the assay measurements, and so this is where GeneLab is awesome, is that they will go through and use a standardized pipeline that a consortium of us at a meeting decided should be the standard pipeline, because there's a whole bunch of different ways that you can run Rna-Seq, but we kind of agreed upon a most common way because there's just not enough time and energy and resources to run it every single different way. So they performed these microarray experiments, and what's cool is they provided all the files for all the steps along the way. So we have their array design reference here, the array data file, their processed data, their raw data, which we're going to use, these cell files. They have graphs that you can look at their processing. So we see a box plot here of their log2, what does this say? Oh, log2 intensities. So you can see like the average intensity across experiment. All this really cool stuff. So if you are to want to do an analysis on your own computer and you're not using the Praxis environment, what we would do is go to these cell files right here, and we would download these. Just click on it and it would save it. You can see I saved them all already. If you're doing this through Praxis, I'm going to have the files already uploaded for you, and I'll show you how to get those. But I want you to be able to know this database and understand it a little bit, so if you want to do additional analyses or something in the future, how you can kind of access this stuff and get the data. So they actually have gone through all the way through and looked at and identified differentially expressed genes. So if you just want the results of the experiment, you can get the raw results. It's kind of raw results is kind of weird, but if you want like the full results outside of the paper, because you're not going to publish 21,000 genes in the paper, like they'll give you the actual data file. So yeah, so that's that. If you want to, you can search by product type, so whether it's a ground experiment, spaceflight experiment, parabolic flight study, what factors, if it was altered gravity, if they were in a cage or not, if it was a cell line versus a real organism, if it was exposed to radiation, all sorts of stuff, different, all these different organisms. You need to know the scientific name. So like Rna sativa, that's rice. Mus musculus is mouse, etc. So you'd have to kind of know Escherichia coli, that's E. coli, Drosophila melanogaster, fruit flies, etc. So I'm not going to go through them all. But yeah, so that's how we access this microarray data. So now if we are in the Praxis environment, what we do, we're going to launch our studio console here. And what I'm going to do is I am going to go to Desktop, Classroom, and then Share. And so this is where I put the data for us to process. I took it from GeneLab and I just uploaded it for you guys so it's easier. And then what you're going to do is Brick 16 is the experiment we're going to do for microarrays. I'm going to click on this right there. And I'm going to go over to More up at the top right there. And I'm going to say copy to, and then I'm going to click right on the desktop again, right here. But this time I'm going to click Classroom and My Files, and then I'm going to name it Brick 16 and hit Save. It should save at that point. I already created a folder to try test this out, but it should save in your files, and so you'll have access to it every time you upload or you start RStudio. So as you can see here in the file section, I have my 6 cell files, and so that's what we are going to use for this analysis. So before we get into the actual RStudio stuff, I'm going to cut the video here so you have time and you don't get behind. And you can make sure all your files, this is our starting point, right, to have all these files saved here. So in the next one, we'll dive into actually doing some of this Rna-Seq analysis. Or sorry, not Rna-Seq, microarrays. We will do Rna-Seq, but not quite yet. So we'll get into the microarray analysis. I hope you can find your files there, and I will catch you in the next one.